So on continuation with IgA, here with we have seen IgD, and it is involved in the production of antibodies by the T lymphocytes, and it is present as monomer, and it weighs only around 1,80,000 Dalton, and it comprises less than 1% of total body content in serum, and it mainly acts as receptor on B cell surface and participates in B cell activation and differentiation. So its IgD molecular weight is 1,80,000 Dalton, and it is the delta type which is having the 70,000 Dalton molecular weight. And IgD is present extremely low concentration. It mainly constitutes 0.2% of the total serum immunoglobulin. And IgD together with IgM, it is the major membrane bound immunoglobulin expressed on the mature B lymphocytes. And there are mainly two subclasses of IgD, that is IgD1 and IgD2, and this plays an important role in the maturation and proliferation of B cells. And IgE is present in the least amounts, very least amount, that is around 0.02% of the total antibody content in the serum, and it is present in the linings of the respiratory tract and intestinal tract and responds to the allergic and anaphylaxis reaction, and this found as monomer the body, and it weighs about 2 lakh Dalton, and its molecular weight is 2 lakh Dalton, and it is epsilon 73,000 Dalton, and this contributes to the allergic reaction, and the allergic anaphylaxis reaction mediate towards the hypersensitivity reaction, and this responsible for the symptoms like hay fever, asthma, anaphylactic shocks, etc. So the main antibody which binds on blood basophils and mast cells, causing the degranulation of mast cells and basophils, and which releases the chemicals called histamine, and this histamine is the chemical which is released into our body, and it is responsible mainly for allergic reactions in our body, and it also provides the immunity against parasites by ADCC, that is antibody dependent cell mediated cytotoxicity. And coming to the antibody structure, these are all the overall structure we have seen. As we have seen IgM as pentameric form, you could see the 5 forms of antibodies present in the IgM antibody, and IgA antibody, it is dimeric form, whereas IgG and IgE, IgD or exist as monomeric forms. And coming to the functions of the body, this antibodies mainly binds to the pathogens of foreign particles, and it activates the immune system in case of bacterial pathogens, and it directly attacks the viral pathogens and assists in the phagocytosis. An antibody provides the long-term protection against the pathogens because it persists for years after the presence of antigen, and it mainly neutralizes the bacterial toxins such as hemolysin, streptococci, the antigen to enhance its efficiency, and it is also acts as the first line of defense for mucosal surfaces, and they ingest mainly by the phagocytosis, and few antibodies which is bind to the antigen that is present on the pathogens. And coming to the investigation and interpretation of methods, ELISA is the only method. ELISA is nothing but the enzyme linked immunosorbent assay. So it is the common laboratory technique which is used to measure the concentration of analyte. So the analyte may be of antibodies or antigens in the solution. So this ELISA technique is mainly used to analyze the interaction of antigen and antibody in the solution. So coming to the principle, it is used as an enzyme to detect the binding of both antigen and antibody, and the enzyme which converts the colorless substrate into colored product, it is called chromogen, and this chromogen indicates the presence of antigen and antibody. And an ELISA, it can be used either the presence of antigens or antibodies in the sample, depending upon how the test is designed. For example, for HIV we used to take this ELISA test, the HIV antigens which is interacted with our antibody, host antibody, and if the antigen antibody reaction occurs in our ELISA test, we used to conclude that HIV is positive through this ELISA test.